So we have Ellie Tyler from the Barts Cancer Institute in London. So Ellie is currently a postdoc researcher at the Barts Cancer Institute at Queen Mary University, and she did her PhD and first postdoc with Dr. Cleo Bishop at the Blizzard Institute, which forms the basis of her talk today. So she will be talking about novel transcriptional regulators of the Senescence program. So there you go. Thank you, Ellie.
Thank you. Thanks, Estella, for that kind introduction, and to the yICSA committee for the opportunity to speak on this webinar series. So hi, everyone. I'm Ellie, and as Estella said, I did my PhD and postdoc with Cleo Bishop at the Blizzard Institute at Queen Mary University of London, which is this beautiful glass building in East London here on the right. And I've now recently moved on to a new postdoc position at the Barts Cancer Institute. So I've left Senescence and I'm now working on the role of the tumour microenvironment in immunotherapy. But I'm going to talk to you today about the work that I did during my PhD with Cleo. So as this is the International Cell Senescence Association webinar series, I'm sure that I don't need to explain to anyone here why senescence is important. But just in case we have any attendees that are new to the field, I'd like to briefly explain why we think it's crucial that we further our understanding of the senescence program. So there's now convincing evidence that senescent cells accumulate in aged tissues and are present at sites of age-related diseases, such as osteoarthritis, atherosclerosis, Alzheimer's disease, and many, many more. And it's thought that long-term retention of deeply senescent cells depletes the pool of cycling cells and stimulates chronic inflammation through secretion of inflammatory factors into the surrounding microenvironment, as I've illustrated with my beautiful green dots coming out of my senescent fibroblast. And this is via the senescence-associated secretory phenotype, or SASP, which ultimately causes aging and age-related diseases. And two landmark papers from the Van Derzen group have shown that selective clearance of P16 positive senescent cells in mice improves both health and lifespan. As you can see in these two chronologically identical mice, the one on the right having had its senescent cells cleared, you see a marked reduction in age-related phenotypes compared to its chronological twin, or littermate, on the left. But despite this growing wealth of knowledge about the relationship between senescence, aging, and age-related diseases, our understanding of the genes driving senescence remains incomplete. So in order to begin to ask questions about the senescence program in my PhD, I first established a model of senescence for primary human mammary fibroblasts in Cleo's lab. And this model was first described in 1961 by Leonard Hayflick and Paul Moorhead. Len, I love to point out, here's the Shrek-like guy in the corner who we all know and love, possibly the grandfather of senescence. So his model involves serial passaging of cells in culture, which leads to a cell cycle arrest mediated by an increase in cell cycle effectors, such as P16, ARF, and P21, as shown here for P16 in green and P21 in red for the primary human mammary fibroblasts that I used for my model. However, the key regulators upstream of these pathways in senescence are still not completely understood. So in this presentation, I will refer to two defined states in the fibroblast lifespan: early proliferating, or EP, and deep senescence, or DS. And EP fibroblasts are serially passaged to senescence and then kept in culture for 21 days further to ensure a stable long-term arrest. So this whole process taking just over 200 days in the lab. And it's at this point that the cells reach what we call deep senescence, and the senescence phenotype becomes really locked in. And we start to see an elaboration of the SASP, which I mentioned earlier, including an increase in inflammatory cytokines such as interleukin-6. And classically, At this point, deeply senescent fibroblasts have been described as irreversibly arrested. So ultimately, we want to ask whether deep senescence is reversible and whether we can use senescence reversal as a tool to identify novel genes driving this senescence phenotype. So Clio has previously published that epithelial cellular senescence, which is solely dependent on p16 expression, can be reversed using p16 siRNA knockdown. And as fibroblast deep senescence is dependent on both p16 and p21 in our model, we hypothesized that transient knockdown of p16 together with p21 would reverse the deeply senescent fibroblasts. So to investigate this hypothesis, I transfected the cells with cyglo control siRNA, p16 and p21 siRNA individually, and then in combination. And then I assessed the impact on numerous cellular and molecular markers classically associated with senescence. So first, looking at DAPI and cell mask, which is a nuclear dye and a whole cell stain, to give you an idea of cell number and morphology. So here we have EP proliferating fibroblasts on the left and the DS fibroblasts transfected with the cyglo negative control on the right. And you can see that the DS fibroblasts have that classic enlarged, flattened morphology. So depletion of p16 with siRNA did not significantly alter the cell number or morphology, but p21 depletion significantly increased cell number and modulated some aspects of senescence morphology towards a proliferative phenotype. But strikingly, depletion of both p16 and p21 together in the DS fibroblasts stimulated an even stronger reversion to a proliferative phenotype, as quantified here on the right with Z-scores in comparison to the cyglo negative control. So we see a significantly increased cell number, decreased cell area, decreased nuclear area, decreased nuclear elongation, and significantly increased nuclear roundness and cell elongation. And further characterization of the reversed phenotype confirmed a significantly increased cycling activity with BRDU incorporation, shown here in green on the left and quantified here for a number of BRDU positive nuclei. And in addition, we saw significantly reduced levels of 8-oxyguanine, a marker of oxidative damage, shown here in green and quantified here on the right. And characterization of the SASP revealed a downregulation of the pro-inflammatory SASP signature, as illustrated by significantly decreased expression and secretion of IL-6. So in summary, we're confident that we have shown that deep senescence is transiently reversible in fibroblasts using siRNA targeting p16 and p21. So we next sought to use senescence reversal as a tool to identify novel genes that drive the senescent phenotype. So using microarray data from proliferating senescent and reversed epithelial cells, we identified genes which increased in epithelial senescence that then decreased following senescence reversal. And an initial siRNA screen in the DS epithelial cells of the top 190 genes which followed this pattern identified 28 siRNAs that strongly induced reversal, as defined by an increase in cell number and the loss of a panel of senescence markers. So to further investigate the relationship between these potential 28 drivers of senescence, I constructed a protein interaction map using the BioGrid database. And from this, 60 genes emerged. And given that DS fibroblast reversal was found to require siRNA knockdown of both p16 and p21, we hypothesized that the drivers identified in the epithelial screen may additionally require knockdown of either p16 or p21 to induce reversal in the DS fibroblasts. So as such, we decided to perform a matrix screen in DS fibroblasts, looking at the 60 experimental siRNAs individually and in combination with P16 or P21 siRNA. So what I wasn't expecting from this experiment was to find an siRNA which was able to reverse the senescence phenotype when transfected on its own, which was why I designed an experiment which ended up investigating 180 different experimental conditions, as well as the respective controls. So it was very much to my surprise that we did find one siRNA which did do that. So this is, well, I'm gonna, in the interest of time, I'm just gonna skip over the other 179 conditions and focus today on early growth response 2, or EGR2, which reversed the senescence phenotype when transfected individually in both DS epithelial cells, as shown here on the left, and the DS fibroblasts. And this is quantified here on the right for cell number and senescence associated cell morphologies. And further characterization also revealed a significant decrease in the expression and secretion of the SASP factor IL-6. So what is EGR2? Apologies for the wall of text, but this is pretty much all that we know in senescence at the moment. It's a transcription factor, and it's a member of the EGR family, which consists of 4 family members, which all share a highly homologous DNA binding zinc finger domains that combine to the same consensus DNA binding motif. And EGR2 has been described as involved in several cellular processes, including cell cycle and proliferation and cell cycle exit. Mutations in EGR2 lead to dysregulation of Schwann cell proliferation, which results in peripheral neuropathies, such as Charcot-Marie Tooth type 1, which is where a lot of the work on EGR2 has been done previously. In addition, a role for EGR2 as a tumor suppressor has been implicated in many tumor cell types, with one paper reporting that expression of EGR2 was decreased in 34 different cancer cell lines across 8 different types of cancer. We know that one member of this family, EGR1, has been shown to play a role in mouse replicative senescence in MEFs, and also RAF-induced senescence in human fibroblasts, which was some lovely work that came out published by Cyril, one of the yICSA committee members last year. EGR1, 2, and 3 have all been shown to regulate transcription in immortalized murine 3T3 cells, which express oncogenic RAS. But to our knowledge, no direct relationship between EGR2 and deep senescence has previously been described. And thus, we sought to explore this finding in more detail. So further investigation of EGR2 in senescence using immunofluorescent staining in EP and DS epithelial cells and fibroblasts, so epithelial cells on the top, fibroblasts at the bottom, revealed a significant increase of nuclear EGR2 foci, as shown here in green, in both senescent cell types, as quantified here on the right. And we also identified an increase in nuclear EGR2 foci during RAS oncogene-induced senescence in IMR90 fibroblasts, as quantified here. And these findings were further supported by remining publicly available mRNA datasets, which found that the abundance of EGR2 increased during replicative senescence in human dermal fibroblasts, in bleomycin-induced, stress-induced premature senescence in BJ fibroblasts, and RAS oncogene-induced senescence in WI38 fibroblasts. And most importantly, EGR2 also increased in vivo in aged human skin, and a whole blood gene expression meta-analysis looking at over 7,000 human samples showed that EGR2 increases with age in human blood. Thus, increased EGR2 levels appears to be a novel marker of senescence across multiple senescence models in multiple cell and tissue types, and a feature of in vivo aging signatures. So we wanted to then unravel the mechanism through which EGR2 may be driving senescence, So first, we identified previously unreported EGR2 binding sites in the promoter regions of ARF and P16, indicating a potential for EGR2 to bind to and regulate expression of these genes. And to investigate this, we performed a luciferase assay for the ARF and P16 promoters in cells overexpressing EGR2, and we found a significant increase in activity for both promoters, as you can see quantified here, confirming EGR2 as a direct activator of both genes. And furthermore, prolonged expression of EGR2 increased ARF and P16 mRNA levels. But importantly, given that all the EGR family members target the same consensus DNA binding motif, we also found in our published microarray dataset that EGR2 is the only member of the EGR family with significantly increased gene expression in DS epithelial cells. And using qPCR, as shown on the left, we found the same is true in DS fibroblasts. And we've also found that EGR2 is the only member of the EGR family capable of directly activating both ARF and P16 promoters. And stable overexpression of EGR2 in normal human HS68 diploid fibroblasts was sufficient to induce proliferation arrest, as you can see in these representative images and quantified here over the course of 18 days. So bringing all of this together, we hope that we've shown that EGR2 acts as a transcriptional regulator of senescence via both the ARF and P16 pathways. And as EGR2 is a transcription factor with the potential to regulate a network of genes in senescence, we would next like to explore the possible mechanisms through which EGR2 may be driving senescence and identify a panel of genes that might be regulated by EGR2 during senescence. So remining of our senescent epithelial cell microarray dataset enabled us to ask whether there was enrichment for the EGR2 consensus binding sequence in genes upregulated in epithelial cellular senescence. And interestingly, there was a small but significant enrichment for EGR2 binding sites at the promoters of genes upregulated in deeply senescent epithelial cells. And quite interestingly, 13 of the siRNAs which strongly induced reversal in DS epithelial cells were identified to target genes which possess an EGR2 binding site. And 9 of these siRNAs also reversed deep senescence in combination with P21 in the DS fibroblast screen. And further data mining to build an interaction map of these genes revealed connections with EGR2 and P16. So we hypothesize that EGR2 may potentially regulate the expression of these genes in senescence, although this is yet to be investigated further. And unfortunately, like everyone, including Pip, during Corona times, we've had to postpone and delay a lot of the experiments. But in the future, we would like to further explore the transcriptome regulated by EGR2 in senescence in order to more completely understand EGR2's role in regulating senescence and potentially identify therapeutic targets for treatment of aging and age-related diseases. Ooh, sorry about that. So to sum up, I hope that I've convinced you that deep senescence is transiently reversible in normal adult human fibroblasts via P16 and P21 silencing, that loss of EGR2 transiently reverses epithelial and fibroblast deep senescence. The EGR2 is a novel marker of senescence in multiple models of senescence, and prolonged expression of EGR2 induces fibroblast senescence. And then finally, that mechanistically binding of EGR2 to the P16 and ARF promoters regulates the senescence program. And finally, I'd like to thank Cleo for her fantastic support as a supervisor during my PhD and postdoc, Mike and my second supervisor, the Bishop Lab, and Ana Gutierrez del Arroyo, who did a lot of the mechanistic work on that final slide, or the second to last slide. Luke, who was the siRNA screening facility manager at the Blizzard, and Rob Lowe, who did a lot of the bioinformatics work. Yeah, and thank you to all of you for listening. And I'd be happy to take any questions. Oh, and this is the lab just before I left in December. So, yeah. Shall I stop sharing?
Great, thank you, Ellie. Very nice talk. So we've got a question from P-Jat. I don't know what that is. Okay. You are mute now. Can you?
Ellie?
Yes.
Yes. Ellie, that was a very nice, clear talk. I have a question. You've made a point of saying that talking about transient expression and transient reversal. What happened if you stably silence EGR2, and then in fact in the deeply senescent cells, will that essentially immortalize those cells?
A fantastic question. Is that Palmer? Yeah, hi. I made a point of saying transient.
Yes, you did.
Because in the method, the technique that we have at the moment, we have used siRNA knockdown, which is of course a transient reversal, a transient knockdown. So we have one of the experiments that we were due to perform just before Corona hit was to put in a stable knockdown of EGR2 into the fibroblasts and see what happens, because at this point in time we don't have that data, so we don't know. But yes, you're right, the possibility is that they would become immortalized or potentially pre-tumorigenic, especially as we know EGR2 is a tumor suppressor.
Great. Thank you very much. Thank you.
Thank you.
Okay, we have another question. Let me see. You're mute now.
Hi, Ali. This is Taranjeet talking from Northern Ireland. Deep senescence obviously is something me and my colleague Andrei Ivanov, who's in QMUL at the moment, I think. I don't know if Andrei is on this talk there, but we showed a lot of chromatin changes that are important in deep senescence. So you have loss of lamins, you have increased macro H2A. Do you have any plans to check for chromatin changes upon deep senescence reversal?
Another fantastic question. So that is something we would love to do, and we would love to look, say, at where EGR2 is binding across the chromatin in deep senescence. Not experiments that we have funding or time for at the moment, and obviously I've moved on from the role, but definitely something that I think would be really interesting to look at, especially given Oliver Bischoff's paper, which came out last week, I think, showing the changes in the transcriptional hierarchy in senescence, I think, and the role of enhancers and the change in chromatin in that. Yeah, I think that would be really interesting to explore, but yeah, not something we have on our books at the moment.
Okay, thank you.
Thank you.
Okay, any more questions? I have one actually. If you were to target this in vivo for like the reversal in vivo, do you know if there is any potential of target, like if there is any cell that particularly expresses EGR2 or?
That's a really good question. The things we know about EGR2 at the moment, people have studied it a lot in Schwann cells and in the peripheral nervous system, but also in T-cell anergy. It's very important in exhausted T cells. So I think you might see an effect on the immune system and on energic T cells as well. And that's actually not something I've thought about. It's not something you'd want to target in vivo anyway, because you probably just end up creating cancer. So I think the idea is that hopefully if we understand more about the network that it regulates, that there will be specific senescence targets that that we can then manipulate.
Okay,
Yeah.
great. Here's another question from Juan Carlos Acosta. I think you can now talk.
Yeah, I think. Hello, very nice talk. I have a question about, do you expect if you do the same experiment with RB, you see like a reversal of senescence? And why is not RB one of the hits that you get in the screening, subsequent screening?
So if I knock down RB, would I see
this is the target of P16 and P21 at the end of the day. So if you just single target RB, would you expect like a reversal, transient reversal of senescence in your model?
It's not something we've tried. I'm trying to recall Beausejour's paper and the kind of senescence reversal papers where they show that knockdown, I think they showed that knockdown of RB reversed senescence.
Yeah.
Yeah. So I think the same would be true in our model. But I think that paper also concluded that the expression of P16 created an obstacle to reversal that they couldn't overcome. So I think the addition of the P16 siRNA with the P21 is a new method for reversing the clot.
So do you expect any other additional function independent of RB or?
For EGR2 or for P16?
No, for P16. Obviously for EGR2, I suspect. Yeah. But for P16 or one of the guys? Or?
No. But yeah, I see your point, because if they're both going through RB, then why wouldn't just knocking down RB do the same thing?
Yeah, I'm saying that maybe there are additional functions. And
we've not looked, but yeah, possible.
Okay. Yeah, very nice talk. Thanks.
Thanks, Juan Carlos.
Okay, we have 2 more questions.
Oh, wow.
I think Dee Bennett, you can talk now.
Hello, Dot.
Hello. Yeah, so just to comment on the previous question, P21 doesn't act only through RB. It can inhibit mitosis directly. It inhibits other CDKs. And P16 doesn't only inhibit RB either. It inhibits the other RB family members, which may be involved in senescence in different cell types. So, RB might not work in your cells. It may just depend on different cell types. So just a comment.
Thank you, Dot. Yeah, absolutely right. What Dot said.
Yep.
Okay, and then last question now. It's again.
Ellie?
Yeah?
This is not a question, this is just a comment. A few questions ago, you mentioned that Schwann cells express EGR2, right? Yeah, so in fact, it was shown about 25 years ago by Hartmut Land that if you actually express RAS in Schwann cells, it actually causes them to senesce. What you might find that if you actually silence EGR2 and RAS, you actually might get the same transform phenotype. So there is a paper from Hartmut Land about 25 years ago. So that might be of interest from that viewpoint. So,
thank you. Yeah, I wasn't aware of that. That's great. So what was the lab again?
It was Hartmut Land, I think, is the last author.
Okay.
And I think Anne Ridley's on there. And they were really, this was in the ideas when they're talking about immortalizing genes plus RAS causing transformation. But it's Schwann cells plus RAS, growth arrest. If you put in like inactivating the RBP53 pathways, the 2 transform. So you might find that silencing or stably silencing EGR2 plus RAS would actually transform them, since that was just a comment.
Okay, right. Thank you.
You're welcome.
Okay, so if we don't have any more questions, just thank you so much, Ellie and Philip, for joining us today, and thank you everyone as well. Hopefully we see you in a couple of weeks for the next seminar session.
Thank you. Thanks, Estella.
Bye, see you.